In previous videos, I talked about cell division and the mechanisms by which cells control growth and division, as well as the details of meiosis and how chromosome number is controlled across the generations through the creation of haploid gametes that have half the genetic content of the germ cells they came from. You probably wondered during that discussion whether mistakes ever happen during that complicated process, and the answer is absolutely. It's not uncommon for mistakes to happen during cell division. And in this video, I'll talk about those errors and their consequences. I'll focus on errors during meiosis because obviously meiosis is that mode of cell division that is generating gametes, so this can have particularly disastrous consequences in the context of human reproduction. Mistakes do happen during mitosis as well, but they are less consequential to the organism. I'll talk about a couple of different types of errors in meiosis. Errors that occur during recombination or crossing over, when DNA molecules are actually breaking and exchanging gene sequences, and errors of segregation, where there's a failure of the spindle checkpoint during meiosis and the chromosomes don't segregate properly. I'll start with recombination errors. The first type of recombination error that I want to talk about is non-reciprocal recombination. What normally happens during meiosis 1, prophase 1, is that the duplicated homologs that were copied in S phase are paired up, and we see a crossover event. Under normal circumstances, that exchange is reciprocal, meaning that there's a specific breakpoint that's introduced in the DNA at the same position on each of those non-sister chromatids, and then there's an equal exchange of equivalent bits of genetic material. But if the breakpoints that are introduced are not exactly aligned with one another, in other words, let's say that on this chromosome we had a breakpoint introduced between these two sequences for genes B and C, but on this DNA molecule the breakpoint is above the B gene. If a crossover occurs in this way, now this arm of this chromatid is attached to this chromosome. This arm of this chromatid is attached to this chromosome. And what we would get, if the first meiotic division proceeded, is a duplication of whatever this gene B is. This chromosome would have had A, B, and then B, C, D transferred from the other chromatid. And in contrast, this chromatid would now have genes A, and then C and D, and would be missing gene B. So what we see is that this chromosome has suffered a deletion of a gene, and this chromosome has now achieved a duplication of this specific gene. What's the consequence of this? Well, it depends very much on what gene B encodes. If it's a gene that's not really subjected to very tight regulation in terms of how much of that gene is expressed and at what time points during development, then this may not be a very important event, and that may be tolerated very well. But if this is a very important, let's say, developmental gene, and the dosage is very important to maintain, then having an extra copy of that gene or a missing copy of that gene could actually be associated with disease. Remember that what's going to happen to each of these sister chromatids is that they're going to go through meiosis 1, and in meiosis 1 we're going to see those homolog pairs separated from one another, and then they're going to go through meiosis 2 where the sister chromatids are separated from one another when those cohesins are cleaved, and so each of these chromatids is going to end up in a different gamete. So if we just write down what genes are going to be present in these gametes, let's say these are eggs, this chromatid ends up in this egg and genes A, B, C, and D are present. This chromatid ends up in this egg, and so this egg has A, B, B, C, D. This chromatid ends up in this egg and only has genes A, C, and D, and this chromatid ends up in this egg. These are ova or eggs, and we know that in the process of fertilization, they're going to be uniting with a sperm cell that is going to ideally be carrying its own set of correct DNA. If it unites with this egg, we can see that that new embryo that's formed is going to be short one copy of gene B. It didn't get one from this maternal parent, but it did get one from the other parent. If this sperm unites with this egg, then that new embryo that's formed is going to have one extra copy of that gene B. So again, depending very much on what that gene B encodes, we could see different consequences as a result of that duplication or deletion of genetic information. In a minute, I'll talk a bit more about this idea of gene dosage effects and gene duplication and how that leads to variation across the genome. and also how that can be associated with the evolution of gene families. But first, I want to discuss that second error, non-homologous recombination. In this case, the wrong chromosomes get paired up in meiosis 1. Again, under normal circumstances, what gets paired up in prophase 1 are the matching sets of homologs. This duplicated homolog that has genes A, B, C, and D should be paired up with its partner homolog that also has genes A, B, C, and D. But sometimes a mistake occurs and a chromosome can be paired up with the wrong partner. For example, this duplicated chromosome with genes E, F, G, and H. You might ask yourself, how can that be possible? Because these sequences shouldn't be similar at all, so how could the cell make a mistake like that? Well, it turns out that an incredible amount of the human genome is made up of repetitive DNA sequences. And when those are matched up, that can make mistakes like this happen. So what would happen if these chromosomes were paired up and then we saw a recombination event between them? Let's say that the breakpoint is here between genes B and C on this chromosome, and then the other breakpoint was between genes F and G on this non-homologous chromosome, and then we saw exchange of DNA. What we would end up with after that recombination was complete is a chromatid that now has genes A, B, G, and H, and another chromatid over here that would have genes E, F, C, and D. This results in a translocation of genetic information. Genes that were supposed to be present on this chromosome have now been moved or translocated to another chromosome in the genome. Notice that this also results in a deletion, because this sister chromatid is now missing genes C and D. And again, remember, these DNAs end up in different gametes, so whichever gamete inherits this particular chromatid is going to be missing genes C and D on this bit of DNA. So when there's an error in recombination, in which non-homologous chromosomes are paired up, and they recombine or exchange genetic information, that's going to result in a translocation and deletion of genetic information. Once again, the consequences of that mistake are going to depend very much on what those genes actually encode. What are the proteins that are produced as a consequence of those genes being expressed? And what are the consequences of not having that protein or having too much of that protein in the cell? How does this play out in terms of evolution? Once you start having these duplication events, you can start to see the creation of what are called gene families. Sets of genes that have related functions that are all descended from one original ancestral gene. Let's say that you have that chromosome and it's got genes A, B, C, and D on it. Let's say that gene B is an essential gene. You absolutely have to have the function of whatever that gene encodes. And if you make mutations that change the function of that gene, then that's a lethal mutation and the organism cannot survive. What happens after that gene duplication event where now you have two copies of that gene? As long as one copy stays with its normal sequence and continues to encode the normal gene product, that organism is going to survive, and that second copy that's the result of the gene duplication event is now free to undergo mutation, which happens all the time, and to possibly acquire new functions. If those proteins become different enough, that can lead to increasing complexity in terms of physiology. And if we see isolation of populations with those different genetic sequences, over time that can actually lead to speciation, where organisms become different enough from one another and populations of organisms become different enough from one another that they actually become separate species. This is really nicely illustrated by the globin gene family. Globins, as we talked about before when we looked at hemoglobin, are the proteins that bind and carry oxygen, and there are dozens of types of globin proteins. But if we go back in time about 800 million years or so ago, there was just one globin gene. This would have been the common ancestor of the family of globin genes that exist today across species. If we look just at the globin genes that are present in the human genome, what we would see is that there are two separate clusters of globin genes, the alpha globin gene cluster on chromosome 16 and the beta globin gene cluster, which is on chromosome 11. Focusing just on the beta globin gene cluster, just for the sake of simplicity, what we would see is that there are 5 globin genes in this beta globin gene cluster that encode functional proteins, and then one pseudogene that does not encode a functional protein. Pseudogenes have mutations that destroy the coded protein's function. So while they might once have been a functional gene, they no longer have any function. But all of these other copies do encode different versions of globin protein. How did we end up with so many copies of these genes? The molecular evidence suggests that at some point there was a duplication event, just like we went over. That could have occurred during recombination. So we ended up with 2 copies of that ancestral globin gene, now referred to as alpha and beta. Over time, those 2 copies accumulated separate mutations, causing them to diverge from one another in sequence, leading to proteins with different structures and therefore different properties. These would have been the ancestral genes to the alpha cluster and the beta cluster. Then a translocation event separated those 2 genes and put them on different chromosomes. This could have occurred through a non-homologous recombination event, perhaps. Now we've got these related genes on separate chromosomes. Further gene duplication events occurred over hundreds of millions of years, leading to the creation of these clusters of genes that have highly related sequences but differ in their functions. They're all globin genes. They all encode globin proteins that are part of the hemoglobin protein complex. But over time, they've acquired different physiological functions. These 2 versions in the beta cluster, symbolized by gamma, are fetal versions of hemoglobin that are only expressed during fetal development. They have a slightly higher affinity for oxygen than the other adult hemoglobin versions that are encoded by the other highly similar gene sequences. And because of that slightly higher affinity for oxygen, the fetus actually has an advantage in survival in being able to acquire oxygen and uptake oxygen and hold on to oxygen, even when oxygen is at slightly lower partial pressures. This is the situation that we see in the human placenta. Where the fetus is acquiring its oxygen, there's actually a mix of fresh oxygenated blood and deoxygenated blood. And so the concentration of oxygen is a little bit lower in that tissue. So the fetus is actually requiring a hemoglobin protein that has a higher affinity for oxygen so that it can bind it at these lower concentrations. And that functional need drove the natural selection of a higher affinity globin, giving human fetuses a very definite selective advantage that has been maintained over millions of years. So gene duplications aren't always a bad thing. They can be associated with speciation and with the evolution of genes with new functions and therefore increasing complexity of the organism. Up until about 20 or so years ago, it was thought that most of the human genome, if we looked at different humans in the population, was actually pretty similar. Maybe we would see 0.1 to 0.4% of the genome varying, and the way that they would vary is in terms of single nucleotide polymorphisms, or SNPs. These are individual nucleotide base pairs that vary between humans because of random mutations that are introduced all of the time. Before the advent of modern, high-throughput sequencing technologies that became available over the last 15 to 20 years, that was our understanding of variation between individual humans. Now, with next-generation sequencing, you can sequence an entire human genome in hours. So thousands and thousands of humans have had their entire DNA sequenced and compared. And what has been discovered is there's a considerably greater level of variation than we ever thought possible. More specifically, there is a ton more gene copy number variation. Not just those slight changes to base pair sequences as a result of mutation, but large-scale duplications and deletions that have led to significant differences in how many copies of particular genes a given individual human might have. This figure shows all of the autosomes and sex chromosomes in one complete set of genetic information. Actually, slightly more than one set because it's got both of the sex chromosomes, X and Y. What these lines are indicating is positions within the human genome where this kind of gene copy number variation exists. Overall, if you compare the DNA sequences of 2 individual humans, they would show copy number variation at about 0.4% of the genome. At a particular position in my genome, I might have 3 copies of a particular gene, and at that same gene locus, you might only have 1 copy of that gene. As you can imagine, depending on the gene, that's going to lead to differences in physiological function, differences in metabolism, and potentially differences in disease risk. And there are a number of human diseases that have been linked to this sort of copy number variation. It's been particularly well demonstrated in human cognitive disorders and psychiatric disorders, things like schizophrenia and autism, which seem to have a common underlying basis in developmental processes that go on in the central nervous system. 0.4% might not sound like a whole lot of DNA, but remember that the human genome contains 3 times 10 to the 9th base pairs, so 0.4% of 3 billion base pairs of DNA is a significant amount of variation. So errors in recombination can lead to both duplications of gene sequences and translocations, which can be associated with disease risk, as well as evolution of new functions of genes and gene families. What I want to talk about next are the other types of errors that we see happening in meiosis, where there's a failure at the spindle checkpoint that regulates the progression from metaphase to anaphase. We already talked about that in the context of mitosis. The very same checkpoint operates in meiosis. The function is the same. It ensures that all chromosomes are correctly lined up and correctly connected to the spindle apparatus before allowing the cell to proceed from metaphase to anaphase. If there's a failure of that spindle checkpoint and the cell allows chromosomes to prematurely be segregated before they're correctly hooked up to the kinetochore microtubules, then what we see is non-disjunction, a failure of correct segregation. This could happen in meiosis 1, where we know that normally homologs are being separated, or it could happen in meiosis 2, where normally sister chromatids are being separated. Let's take a look at what would happen in either of those scenarios. Here on the left, we have an example of a meiosis where the non-disjunction event occurs in meiosis 1. On the right, we see anaphase 1, where this tetrad is being correctly separated, one homolog being sent to this pole of the cell, the other homolog being sent to that pole of the cell. But this tetrad on the left is not being segregated correctly. There's a non-disjunction event, a failure to disjoin, where the whole tetrad goes in this direction. And so what we get at the end of that first meiotic division is a cell that has one too many of this set of duplicated homologs. It inherited too much DNA here, and this cell, which didn't inherit any copies of that duplicated homolog set, is missing genetic information. If these two cells then undergo that second round of meiotic division, where sister chromatids are now separated from one another, then what we end up with is half of those products of this meiosis that are N+1, meaning they have one too many chromosomes of whatever that type of chromosome was, and half of the gametes produced will be N-1, where there are gametes that are missing that entire set of genetic information. What's going to be the consequence of that? It depends on what that chromosome is. But for most chromosomes, that's not going to be tolerated, and this will be a lethal event. What if the failure to segregate occurs in meiosis 2? Let's say meiosis 1 proceeds normally. These tetrads are both correctly separated, with duplicated homologs being sent to opposite poles of the cell. This cell on the right is shown undergoing a normal meiosis 2. Sister chromatids are appropriately separated, and from this lineage we get two gametes that are entirely normal and haploid. They have one of each type of chromosome as they should. But let's say that this cell on the left has a failure to segregate one of its chromosome sets correctly, and so these two sister chromatids, instead of being separated in meiosis 2, both end up going to the same pole due to a failure of the spindle checkpoint. Both of these gametes would be abnormal, one N+1 and one N-1. When you have a non-disjunction like this and you end up with too many or too few chromosomes, that's a defect in chromosome number, which is called aneuploidy. Remember that ploidy refers to chromosome number. U means true, so aneuploidy means not true chromosome number. What would be the consequences to a human if this happens? For most of the autosomes, aneuploidy is not survivable. If you look at human pregnancy and you look at the rate at which a given human pregnancy is going to survive past the first trimester, what you find is that about 20% of pregnancies spontaneously abort in the first trimester. A huge percentage of those actually fail in the first 2 weeks before the woman even knows that she's pregnant. So there's a huge failure rate associated with the beginning or onset of pregnancy. About 90% of those spontaneous abortions are associated with fetal chromosomal abnormalities, some sort of aneuploidy or other chromosomal abnormality that prevents that embryo from making it further into fetal development. Most autosomal aneuploidies are going to be lethal. They're not going to be survivable. You're not even going to make it to a live birth, much less make it to adulthood. But there are 3 that are actually seen a bit more often and do make it to a live birth. These are trisomies, tri meaning 3, so 3 chromosomes instead of 2. And those are chromosomes 21, 18, and 13. The incidence of these trisomies for chromosome 21 is about 1 in 1,000 overall, about 1 in 5,000 for chromosome 18, and about 1 in 10,000 for chromosome 13. Here on the left is the normal human karyotype that we looked at before, where we see the paired duplicated homologs for all of our sets of autosomes 1 through 22, and then one set of sex chromosomes, X and Y. And on the right is a karyotype that's depicting a trisomy 21, where this individual has inherited 3 sets of chromosome 21. This is associated with Down syndrome, a pretty common human genetic disorder. Down syndrome is associated with cognitive disability, as well as various developmental defects in other organ systems, including the cardiovascular system. Kids born with Down syndrome very often have holes in their heart that have to be repaired, or other heart defects that have to be repaired after they're born. It's also associated with defects in the digestive tract. They have a problem with getting adequate nutrition, and they have a whole host of other challenges as well. If you look at the incidence at which Down syndrome occurs as a function of maternal age, you see something very interesting. Here on the y-axis is incidence of trisomy 21 per 1,000 live births. So this is babies born. And down here on the x-axis is maternal age, going from onset of puberty about age 15 on up to age 45. What you see is that up to about age 25, incidence of Down syndrome is pretty low, about 1 in 1,000-ish. But if you look at the incidence of Down syndrome as mom's age goes up, then by the time you get to age 45, what you see is that the incidence of Down syndrome has gone up hugely and is about 1 in 25. To understand why that happens, you have to understand a little bit more about how the meiotic divisions that produce the maternal gametes happens in humans, because the specifics are a little bit different than what we saw with our general meiosis discussion. In humans, the entire set of gametes, or oocytes, is produced during female fetal development. So every woman's entire set of eggs, around 400,000 total, was established when she was still a fetus in her mother's womb. All of the primordial germ cells that generate those oocytes in the female fetus initiate meiosis before birth, but then that meiosis stalls at prophase 1. Every one of those primordial germ cells have initiated meiosis and then stalled at prophase 1 with the duplicated homologs all paired up in synapsis. That meiotic division then just pauses. It doesn't go any further. This fetus continues development. It's born, it goes through childhood, it goes through early adolescence. Then, beginning with puberty, at every menstrual cycle there's an ovulation event, where an oocyte matures and is released from the ovary. That oocyte that is being ovulated reinitiates meiosis from where it stalled. So we see meiosis 1 reinitiating as that oocyte travels down that fallopian tube. But this oocyte will only complete the second meiotic division if it's fertilized by a sperm cell. So it's a really complicated meiotic division that human females go through. A human female that's 25 years old and ovulating an oocyte that is going to be fertilized by a sperm is releasing an oocyte that actually began meiosis 25 years previously when she was still a fetus in her mother's womb. Likewise, a 45-year-old woman is releasing an oocyte that initiated meiosis 45 years ago. And at that point, this very complicated molecular process that was initiated in the fetus during fetal development and has all of these proteins that are connecting these tetrads up and the kinetochores and all that good stuff, we're asking that whole molecular complex to remain intact and then at ovulation to pick up and begin where it left off and reinitiate meiotic division. As you can imagine, proteins degrade over time, and this molecular complex could fall apart in any number of ways. So when we think about this increased incidence of trisomy 21 as a function of maternal age, that's because there's a huge increased incidence of non-disjunctions, or failures to disjoin, as a function of maternal age in human females. The molecular details of what exactly goes wrong and what happens to this protein complex that's holding these together and why they don't segregate appropriately the way they're supposed to is not entirely understood. We don't know the details of why that happens. We just know that it does. So you might ask yourself, well, does the same thing happen in human males? Is paternal age actually associated with any sort of increase in genetic defects and genetic disorders in the offspring? And the answer is maybe, but not in the same way. To understand why, we have to look at how spermatogenesis occurs in human males, because it's very different from what happens in females. Whereas human females get their primordial germ cells and their whole collection of oocytes established before birth, and that's all they're ever going to get, ever, in males it's very different. Males start with a primordial germ cell and expand that out to a population of stem cells that exist in the testis. Then, at puberty, those stem cells undergo characteristic sets of division to renew and expand the population of stem cells. As each of those stem cells undergoes a differentiation event, leading to mature spermatozoa, each of those cell divisions is associated with a DNA replication event. And every time DNA gets replicated, there are mutations that get introduced into the genome, because copying DNA is not a 100% accurate process. So when we're thinking about this stem cell population that maintains this set of cells that are constantly capable of dividing and producing new spermatozoa, millions and millions of them every day, those cells actually replicate and divide about 23 times a year to renew this population of stem cells. As those stem cells undergo divisions to produce the mature sperm, that's associated with another 4 replication events. Over time, as each spermatozoa is produced, that's the result of hundreds of previous cell divisions, and each of those rounds of DNA replication introduces new mutations in the form of single nucleotide base pair changes in that genome. That's the genetic payload being carried by that sperm. So if we compare the level of mutation that's present in a sperm to the level of mutation that's present in oocyte, in sperm we see many, many more mutations. In contrast, in females, once those oocytes are established, they don't divide again. So throughout the woman's life, those are not undergoing additional rounds of division. Only in the males do we see that happening. The types of genetic disorders that you see with a higher prevalence in older fathers are the result of those kinds of single nucleotide base pair changes. There are disease syndromes like achondroplasia and Apert syndrome that have very well established associations with increasing paternal age, where you can see that as dad gets older, those disorders become much more likely to occur. There are other disorders that are also associated with increasing paternal age, for example, autism. Aside from trisomy 21, which is relatively common, aneuploidies of the other autosomes are not well tolerated. Part of the explanation is how many genes are on the autosomes, as well as what types of genes are on those autosomes. For example, chromosome number 1 is humongous and contains 4,500 individual genes. If you inherit an extra copy of chromosome 1, you are dead very early in embryonic development. That's just too many genes in extra copy to be tolerated during embryonic development, when there are still going to be lots and lots of genes on that chromosome that are going to affect development in an adverse way. You can also have other chromosomes that are smaller. They don't have as many genes, but may have very, very important genes that cannot be tolerated in too many copies or too few copies. In other words, where changes in gene dosage aren't allowed. As it turns out, aneuploidies of the sex chromosomes are much more common and are better tolerated in terms of physiological function and development. If we look at the karyotypes that are associated with sex chromosome abnormalities, we can see that a couple of the most common are where we have one extra copy or one too few of the X or the Y. The first one that's listed here is Turner syndrome, where the individual only has 45 chromosomes, so they have one X, but they're missing that other sex chromosome, either an X or a Y. That's often referred to as being XO. The incidence of that is about 1 in 2,000 female births, and for the most part, these folks are okay. They don't have cognitive disability, they don't have a whole lot of behavioral disorders associated with that, They do have a slight defect in gonadal function, so reproduction is impacted in these individuals, and they have some other kidney and cardiovascular defects. But in general, they're not too severe, and they definitely survive past birth and into adulthood just fine. XXY leads to Klinefelter syndrome, and that occurs in about 1 in every 576 males. These folks have a little bit of cognitive disability, a little bit of behavioral changes, taller than average, hypogonadism impacting fertility, and various disorders in other organ systems, but again, they survive just fine. There are other trisomies where we've got extra copies of the Y or the X occurring in various frequencies, and we even see tetraploidies or pentaploids, where you've got 4 copies of sex chromosomes, 5 copies of sex chromosomes. They're pretty rare, but they do survive. So why are these aneuploidies better tolerated than aneuploidies of the autosomes? 2 reasons. First, it turns out that in order to compensate for the fact that females have 2 X chromosomes and males only have 1 X chromosome, what typically happens is that chromosomes in excess of 1 get inactivated during development. Random inactivation of all but 1 X chromosome happens. So if you have extra copies of X's, then those extra copies just get inactivated. It doesn't work perfectly, and so that's why we see some disorders associated with having those extra X's. Second, extra copies of the Y chromosome can be tolerated because the Y is very small. It doesn't have many genes, and there aren't any essential genes on the Y. That makes sense if you think about it, because half the human population doesn't have a Y chromosome, so there can't really be any physiologically essential genes on it. It does have one very important gene, and that's SRY, the gene that confers maleness, which we'll talk about more in our genetics discussion. But in terms of essential physiology, there really aren't any. There are only about 200 genes total on the Y chromosome, and only about 70 of those encode proteins. So if you have extra copies of the Y, it's not really a big deal. But these are really the exceptions to the rule. Most chromosomal abnormalities are going to be involving the autosomes, just because there are 22 of those and only 1 set of the sex chromosomes. And most of those are lethal and not tolerated well at all. In this video, we've seen how mistakes can occur during meiosis and how those can be significant in terms of evolution of populations as well as for human disease. Now that we understand how reproduction occurs at the cellular level, we'll turn our attention to genetics.